Hello, everybody. Good morning, good evening, good night, whenever you're listening to this recording. I just want to welcome everybody. We are going to be talking about chapter 18, which is our chapter on blood. So, this is the lab portion for our Bio 211. I want to welcome you to the next chapter here. We get the opportunity in this video to start talking about one of our connective tissue types. And so let's delve right into it and start talking about blood, what it is, what it does, and some of the cool characteristics that we're going to learn about it. So, folks, just to let you know, like I said before, blood is one of our three types of connective tissue. It fits in the last category that we learned about in chapter 5 when we were learning about tissues, which is fluid connective tissue. And the term that you see on the screen there, it says it's continuously regenerated, and that is very true, right? Blood is constantly being added to, and it's constantly having things taken away from it. For example, a red blood cell lives for about 120 days. So about every 4 months, you are going to constitute, all right, some new red blood cells to replace old or damaged ones. So the purpose of our blood, the connective tissue, the fluid connective tissue, as you can see here, is to move gases, nutrients, waste, and hormones. Keep in mind, I like to think of the blood as our railway system of our body. We need to transport things, any of those that I just mentioned there, gases, nutrients, waste, and hormones, from one place to another. And one of the better ways to do that is through the circulatory system here. And so the circulatory system is part of the cardiovascular system. The cardiovascular system includes our heart. The heart pumps the blood, and when the blood exits the heart, it exits through the arteries. So definition of an artery is a blood vessel that transports blood away from the heart. The definition of a vein is a blood vessel that transports blood towards the heart or to the heart. So arteries carry blood away from the heart, veins carry blood towards the heart. And then we have the area of exchange, and I want you to really commit this to memory, that would be our capillaries. Our capillaries are the blood vessels, the smallest, thinnest blood vessels in our body. And so they're the exchange site. So they're going to allow substances to exit from your bloodstream out into tissues there. And then it's going to, of course, then draw material from your tissues into the blood. So usually nutrient respiratory gases like oxygen will exit out of your blood, and then some of our our respiratory waste gases, which would be like carbon dioxide, are going to exit from the tissues into your bloodstream and circulate back to your lungs to be exhaled. So keep that in mind when we're talking about blood. Blood needs a mode of transportation, or I don't want to say a containment vessel, but a containment unit, which is going to be the cardiovascular system, the circulatory system. So when we look at our blood, we're going to notice that our blood has two major components. All right, we have our blood-formed elements, and then we have our plasma, which is the liquid portion, essentially. So the formed elements are going to be made up of three cell types: erythrocytes, known as red blood cells, or RBCs. And so their main function is to transport respiratory gases like oxygen and carbon dioxide to and from our tissues. Leukocytes, which are our white blood cells, right? These are going to be the cells that are going to protect us from microorganisms or pathogens, viruses, bacteria, hemolysis, microorganisms, and whatnot. And so the white blood cells help to mount the immune response. And then we have our platelets, also known as thrombocytes. And these structures help with the clotting process for the blood. And so essentially it'll prevent blood loss. The other component to our blood is our blood plasma, that is the fluid portion, right? And so we're going to discuss here in this chapter, right, what constitutes or what makes up the fluid portion, the blood plasma, but it's going to obviously be made up of a large portion of fluid, which we'll talk about, and then some substances that are dissolved into that fluid and some that aren't dissolved. So those are going to usually be like plasma proteins and some of our dissolved solutes, which we'll talk about here in a little bit. So the next part of the bloodstream that I want, or blood, is going to be the physical characteristics, what you see when you're looking at blood. So the first one, of course, is the color. And so the color has to do with how much oxygen is actually in your bloodstream. All right. So the degree of oxygenation is going to determine the color of your blood. So blood that has more oxygen is going to have more of a bright red appearance to it. And then blood that is what we call poorly oxygenated or has a lower amount of oxygen, right? That is going to have more of a darker red. Some people might even say closer to a purplish content there. But point being is brighter red, a lot more oxygen, darker red, less oxygen. Normally in adults, you will see, all right, about 5 liters of blood, and it varies depending on the size, the age, and the sex of the individual. But on average, it's about 5 liters of blood when we're talking about our blood volume. So anywhere between 4 to 6 liters there. And like I said, it varies depending on certain characteristics of the individual. The next physical characteristic is viscosity, thickness, right? And so you probably heard that term that blood is thicker than water, which is true. It's literally true, right? Blood is about 4 to 5 times thicker than water. And a lot of this has to do with, all right, the amount of substances that are dissolved or actually just in suspension in our fluid portion of the bloodstream there. So we'll talk about that here in a moment, like what some of those substances are. But if you have more solid material or dissolved material in the fluid and less material, the ratio there swings in favor of the dissolved substances and the suspended substances, you're going to have a thicker blood. If you have a larger amount of fluid in the blood and a smaller amount of dissolved or suspended substances, then that blood won't be as thick and it'll be less viscous, more fluid-like. So, for example, if you increase the number of erythrocytes that are present, say you have something going on with your red bone marrow and it starts kicking out more red blood cells, that in itself can make your blood thicker. And then also, if you also want to increase the thickness of the blood, folks that are dehydrated, for example, right? If you have less fluid, because a large component of your blood plasma, about 92% of it is water. And so if you have a smaller amount of water in there, then the ratio between the actual fluid, like I said before, and the dissolved and suspended substances will swing in favor, all right, of the more solid material. So you'll have a thicker, more viscous material there. So that's viscosity. Onwards to the next physical characteristic of blood, which is the plasma concentration of the solutes, right? Like what's in the liquid, what's suspended, what's dissolved, all right? So the plasma concentration of solutes, and you can see some of the examples up there on the screen, all right? Proteins and ions, for example. Way back when, if you took 211 and we discussed a little bit of some of the characteristics of movements, plasma membrane transport, for example, in chapter 4, we learned about one of the processes called osmosis. And osmosis has to do with water. So if there's a topic or something that you're reading about and it's talking about plasma membrane transport and it doesn't mention anything about water in there, you're not dealing with osmosis. So we have to deal with osmosis. So the solution or the plasma concentration of these solutes is going to help determine in which direction water is going to move, whether it's going to move into the capillaries or whether it's going to move into the tissues from the capillaries. So the example listed here talks about dehydration. So when you're an individual that's dehydrated, right, and we learned about the different tonicities of solutions, again, back in chapter 4, hypertonic solutions, hypotonic solutions, and isotonic solutions. If you don't recall those, watch my video on chapter 4, right? But when you are dehydrated, right, your plasma concentration for the solution is going to be hypertonic, which means there's lots of solutes there. And so here's what happens, and I've mentioned this before in other videos, water is a follower, and it likes to follow solutes. The definition of osmosis is the movement of water from an area of high concentration to an area of low concentration through a semi-permeable membrane. But one of the things too that water likes to do is it likes to move towards the area, all right, that has a higher solute concentration. So if your blood plasma has a higher solute concentration than the interstitial fluid or the ICF, which is the intracellular fluid, then water is going to get drawn out of your tissues and move into your blood plasma, right? Because it's going to move towards the area that has a higher concentration of solutes. So again, we'll talk more about this in later chapters when we talk about fluid balance, but keep in mind, right, what you're going to see is, think of it like this. Water moves from an area of high water concentration to low water concentration. But at the same time, that means it's normally going to move in the area that has the higher solute concentration, which is what we see in dehydrated individuals in their blood plasma. They have less fluid in the blood, higher solute concentration, so therefore water gets pulled out of the tissues towards the blood. Okay, next physical characteristic is going to be temperature. All right, and this is all we know this, and especially if you're watching this video during a wintry day, which today right now happens to be in South Carolina, a cold wintry day. I think the high today was 29 degrees. It was well below freezing, and now it's snowing outside. So it's cold. So one of the things that we're concerned about in cold temperatures is heat. Well, your blood is about 1 degree Celsius higher than your regular normal body temperature. So we all know 98.6 is the regular body temperature. So around 99, 99.6, anywhere between there, is going to be 1 degree higher. And so this is why when you are outside and you walk in from being outside into your house, for example, your blood is now going to move from your core, because that's usually what happens. Blood retreats from your extremities to your core to keep your vital organs warm in colder temperatures. We see this with hypothermia, for example. Well, when you walk in from the outside back to your home, then the blood that was in your core can now move back into the extremities. And as it does so, it helps to warm your extremities up. You might notice that, right, as you start to recover from that cold temperature from being outside. Okay, another physical characteristic of blood is going to be our pH, our blood pH. And we talked about this a lot in 210 in my classes, when we talked about when we were discussing homeostasis. And the normal blood pH for individuals is going to be 7.35 to 7.45, so around 7.4, which is not neutral, but it's slightly alkaline or basic there. Right? And so your body, just like with most systems, wants to keep your blood pH between those two values of 7.35 and 7.45. Otherwise, if you start to swing in either direction, normally if you become more acidic, which is what we're accustomed to do, okay, we can start to cause certain malfunctions in certain molecules. For example, one of our macromolecules, the proteins there, if we start to dip down into a lower pH, right, we become more acidic, we risk what we call protein denaturation, which essentially means, all right, some of these proteins that have been folded up like an origami sculpture by design starts to unfold. And when they lose their shape, they can lose their function, and then they're pretty much useless to us. So that's why we try to maintain our blood pH in that value range of 7.35 to 7.45. All right, so moving on, let's talk about in more detail the components that we find in our blood. Now, earlier on in this video, I told you that primarily our blood is made up of formed elements in the blood plasma. So we're going to now discuss a little bit more in detail what that all is. All right, so here's what's happening. You go into your doctor's office, medical care practitioner's office, and they say, hey, we're going to draw some blood on you. And then we're going to run, a blood panel and see what's going on. So the first thing that they do is they draw blood off of you, okay? And we refer to that as whole blood because it's got everything in your circulatory system, the blood formed elements in the plasma. And they're going to stick it into a device called a centrifuge. Essentially, this is like a big spinning wheel device. And so they'll stick it in the centrifuge and it will spin around, I want to say roughly for about 10 minutes or so. And it's going to start to separate out, right, parts of your blood, namely your formed elements and the blood plasma. So here's what we're going to see. At the very bottom, I'll show you a quick picture and I'll promise you I'll come right back to this slide here. Here you can see a picture here of the blood plasma or the test tube with everything separated out. So at the bottom, you have this red area, then just above it, you've got this kind of gray area there. And then above that, you got this straw colored fluid area here. All right, so we're going to talk about what those three areas comprise or consist of. So at the very bottom, that's going to be your red blood cells. We just talked about what gives your blood its red coloring characteristic, and that's the oxygenation of the blood. So the erythrocytes are going to be on the bottom, and they are going to comprise about 44% of the overall volume of your blood. Right, just above that, and you'll see in the picture, I'll go back to it here in a second, you have a very thin layer, right? And this kind of grayish white layer is called the buffy coat. That's going to contain the remaining blood-formed elements, which are your white blood cells known as leukocytes and your platelets, all right, the thrombocytes. Those are going to be the structures, all right, that are going to be responsible for blood clotting to prevent blood loss. So your remaining blood-formed elements will be in that layer. And that layer is very small. It's about 1%. So far, we've covered about 45% of your blood volume. The remaining 55% sits on top, and that is your blood plasma. And that makes up the remaining 55% there. So if I just switch to this picture here you can kind of see what that will look like in our test tube. And you can kind of see how we're drawing blood on the left side of the screen there, right? Blood being drawn down the vein, stick it in the centrifuge, we spin it down, and then we look at our test tube sample now. Now, if you look to the right of the screen, we can see what's in each of those layers. The easiest one is go to the bottom, and you'll see in that red layer, those are the erythrocytes. And like I said, that makes up 44% of your overall blood sample there. Then just above it, you can see the buffy coat, and that's going to contain your platelets and your white blood cells. And if you look at it, there's not a lot, all right? And we'll talk more about, especially with the white blood cells, most people think that a majority of their white blood cells are in circulation. They are not. They are actually in tissues, thank God, because if you're looking at that blood sample, less than 1% doesn't seem like very much. And of course, everybody wants as many white blood cells as they possibly can get because of how it protects us from the pathogens there that we're exposed to. All right, but more on that in a later chapter. But just keep in mind the buffy coat, very small, about 1% or less, right, is going to contain the remaining blood-formed elements. And then finally, at the top there, you can see we have our plasma. And if you notice, right, we have three subcategories for what's in the plasma: water, proteins, and then other solids. Notice that water is 92%, right? That's quite a bit. So the remaining 8% is going to be comprised of proteins and then our other solids. So we'll go through some of these structures here, what, the proteins and the other solids. But if you look, our proteins, we have albumins, globulins, fibrinogen, regulatory proteins, which we'll get into. And then our solids is pretty much just everything else. And you'll see there electrolytes, so that includes sodium, magnesium, potassium, for example, nutrients, okay, glucose, amino acids, fatty acids, glycerol, respiratory gases. Now, a good portion of the respiratory gases will travel on the erythrocytes, but not all can fit on there. So some will actually be in our blood plasma, and then waste products like nitrogen, urea, and whatnot. All right, so you can kind of see that the blood plasma contains a diversity of different substances, whereas the other layers there just have pretty much some very minimal basic components there. So one of the characteristics when we're looking at centrifuge blood is this term called hematocrit. Right? And so hematocrit is, when we're looking at it from a clinical point of view, for example, like when you go in and get your blood drawn, and then there's a report that's printed out on your blood, and it talks about hematocrit. Basically, hematocrit is going to, on that report, pretty much pertain only to the erythrocytes, right? But keep in mind, hematocrit is a percentage of volume of all formed elements. But go back to this picture here. Check it out. See, less than 1% of your overall blood, but less than 1% is going to be these blood formed elements. The majority of the blood formed elements are going to be the erythrocytes there. So that's the reason why with hematocrit, for the clinical definition, we only pretty much pay attention to the erythrocytes there. So if you look in adult males, it's 42 to 56%, and females, it's 38 to 46%. And you can see below why that is, all right? Males are going to produce, because of the androgen testosterone, more erythropoietin, or EPO. And if you recall what erythropoietin is, you will conclude, all right, erythropoietin is the hormone that stimulates red bone marrow to make more red blood cells. You make more red blood cells, you're going to have a higher percentage of erythrocytes, therefore you'll have a higher hematocrit, and therefore you can see why males have a higher hematocrit than females. All right, so one of the other things that will happen when you get your blood drawn is that the lab will actually create a blood smear. And so that is something that we were going to try to reproduce in our labs, right? But more on that in actual lab class. But a blood smear is essentially, we're going to take a small, very slight, and I'll show you here in a moment here, all right, how we do it. But we're going to take a very small amount of blood and put it, all right, onto a slide, and then we're going to look at it under a light microscope there. So when we look at our blood smear, we're going to be looking primarily at the blood-formed elements there. So the erythrocytes, the red blood cells, and the white blood cells. So obviously, the erythrocytes should be the most numerous because they make up 44% of our blood volume there. And so the cell itself, the erythrocyte, right, will have a reddish color to it, or a pinkish color. And then it looks almost like a donut, except it doesn't have the center missing. I hate when it does that. All right, so there's an erythrocyte. In the center, it'll look kind of pale in the middle there because it's thinner there. So when you look at the erythrocyte from the side, it's going to look like this. And so you can see here in the middle, it'll be more pale. And that's because it's thinner. All right, and keep in mind, I'll tell you now, erythrocytes, mature erythrocytes, are anucleated. They do not have a nucleus. And we'll talk about that in lecture. So the white blood cells, the leukocytes, right, we'll see those. They won't be as numerous as the erythrocytes, but they will be larger. And depending on which type of white blood cell or leukocyte we're looking at, right, we're going to notice the nucleus, all right, different characteristics for different cells. Right, the nucleus can be variable. For example, you can have a multilobular nucleus. So a nucleus can have multiple lobes to it, for example, or it could be just one big nucleus. Right, more on that in lecture. And then finally, you'll see on the blood smear, small little like chunks of something. All right, in this case, they're not just something; they're platelets. Right, platelets are what we call cellular fragments because they break off from a cell called a megakaryocyte that lives in the bone marrow, and just parts of that cell just bust off into circulation and circulate throughout your body. So these structures will be much smaller than the other two blood-formed elements that we talked about. So they'll be smaller than the erythrocytes and the leukocytes there. So here you can see how we make a blood smear. Right, so in this case here, if you just want to do solely a blood smear, you're going to have to do a finger prick on somebody. But if I've already drawn the blood from you, I can get the blood from my blood smear from that sample that I took. So you prick the finger in this case. You can use a glass pipette, all right, and then you can dab it onto the blood and then put it onto your glass slide. And just like it says here, I won't read it off to you, all right, but you just spread it out on the glass slide, put a cover slip on it. Right, prior to putting the cover slip on it, you will use a stain to stain some of these structures here, and then slide it under the glass microscope there, or the light microscope. And so here you can see to the right on our screen, right, what a normal blood smear should look like. And you can see how numerous the erythrocytes are. And you can look at the cell characteristics and notice that the center of those cells are pale in comparison because they are much thinner. And then those bigger cells that look blue purplish, those are the leukocytes, right? And those are your white blood cells. And then you'll see these smaller little fragments of cells, right, throughout the sample. This one here doesn't have very many, but you can see where that one circle is on the lower right side of that picture. You can see one of the platelets there. All righty. Let's talk a little bit about blood typing. Okay, the last part that I'd like to discuss with you folks in regards to the blood chapter for lab are going to be the erythrocytes specifically, but about blood typing. Make sure I got you on the right slide here. Okay, so a couple characteristics and a couple concepts that I want to go through with you. And again, just bear with me. And if it seems like I'm speaking, in very simple terms, it's because I'm trying to, because this is a topic that folks can get easily confused about. And sometimes I even confuse myself. All right. But that's not uncommon because I do that all the time with myself. Okay. So let's talk about the blood types. And I really strongly encourage everybody to at least learn what their blood type is. All right. And it's good to know in case you do actually need to give that information to somebody for whatever purpose it may be. You should just know your blood type. So when we talk about blood type, we're referring to a group of what we call glycoproteins that sit on the plasma membrane surface there. And so we refer to, all right, these glycoproteins, these molecules, all right, as surface antigens. And antigens, all right, in definition can be proteins or carbohydrates, all right. But in this case, when we talk about our blood types, these surface antigens, these glycoproteins, which are going to be found on the surface of our red blood cells, right? We have a system that we categorize them. So you can have, all right, looking at your blood groups, and you can see here on the screen, you have 4 types: type A, type B, type AB, and then type O. So depending on what type of surface antigens you have will determine your ABO blood group. So how do we do this? We are going to see if you have a presence or an absence of 2 types of surface antigens, A antigen and B antigen. All right, so if you are type A, for example, that means that the only surface antigen that you have is antigen A only. So when someone says to you, hey, I am type A, you immediately should be thinking, oh, their red blood cells have the antigen A have the surface antigen A only. So therefore, if you're type B, same thing. You only contain the surface antigen B only. Therefore, if you are type AB, then you'll have both antigens. So both antigen A and antigen B. And if you're type O, you will not have either one. How I remember type O is think of 0. Okay, that means you have 0 surface antigens. So let me repeat myself. Type A means you have surface antigen A only. Type B means you have surface antigen B only. If you are AB, then you have both antigens. And if you are type O, you have neither. O, 0. Okay, so that brings me to the next part of this concept of ABO blood typing. And now that has to do with what antibodies are. Okay, so an antibody is part of your immune system. They are proteins, all right, called depending on what type of response is going to occur. We will learn about these specifics when we study the immune chapter in chapter 22. But for right now, just know, okay, a person's antigen status determines their antibody status. So let's look at this. An anti-A antibody will react with a surface antigen A. Anti-B antibodies will react with surface antigen B. So you see here in this picture, I am type A. I'm A positive. We'll talk about what the positive means, but I am A positive, which means that my red blood cells contain surface antigen A. And what did I say on the previous slide? Anti-A antibodies will react with the surface antigen A. That means that my surface antigen A my surface antigen A reacts with anti-A antibodies. But if you look at this chart here, let me get rid of that crazy line there. If you look at this chart here, you'll notice, I'm only going to talk about me. A type A person, me, since I have the surface antigen A antibody, since I have the surface antigen A glycoprotein on the surface of my erythrocytes, my blood plasma carries anti-B antibodies. So I carry antibodies that will react with a different surface antigen. Think about it. It would be insane, I wouldn't say insane, but it would not be good for us to carry antibodies that react with our own surface antigens, because then our own antibodies would be going after our own red blood cells, and it would cause hemolysis, and we could potentially die. So I just wanted to point that out. So this part right here, it says a person doesn't have antibodies for their own surface proteins. And I just said the reason why. Because if we did, then those antibodies would react with our surface proteins, damage our cells, and then called hemolysis and cause our cells to break apart. But notice below it here, a person does have antibodies to antigens that are foreign to them. So if you see here on the screen, type A blood has anti-B antibodies in the plasma. That's this right here, this whole column here. Type A blood has, the surface antigen A on the red blood cells, but notice the antibodies are the anti-B antibodies. So they won't react with their own erythrocytes, but guess what? They will react with type B blood, and they will react with type AB blood. Because here you can see, look, the anti-B antibodies have that specific configuration that can match up on these glycoproteins on this cell and on this cell, but not this cell, because they don't fit. It's like you have a key, but it just doesn't fit the lock. And then notice type O doesn't have any antigens. Okay? But these people possess both anti-A and anti-B antibodies. Oh, and I'll talk a little bit more about this. Let me go back to this slide here. Okay, so just to review, type A blood has anti-B antibodies. Type B blood has anti-A antibodies in its blood plasma. AB has neither antibodies, but AB has both A antigen and B antigen surface glycoproteins. Type O blood has both anti-A and anti-B antibodies in its blood plasma, but guess what? Type O blood does not have any surface antigens. So it does not have the surface antigen A, or it does not have surface antigen B. So this chart really will help you, I would say, if you're still having some issues, okay, with the conceptualization of it, memorize it for now. All right, and then just kind of reread this slide a couple times. It will make sense to you, I promise you. Trust me. All right, so you can look here and you'll see, right, just keep in mind, okay, a blood type will not carry antibodies for its own surface antigen. Otherwise, you destroy your own cells. Type AB blood has both surface antigens, but it does not contain any antibodies. Type O blood does not have any surface antigens, but it contains both types of antibodies, anti-A antibodies and anti-B antibodies. So this, of course, is going to bring me into that positive and negative portion of our ABO blood typing. So when we talk about, like I said, I'm A positive. Okay, so now we're going to talk about what that positive means. So that positive has to do with what we call the Rh factor, also known as the surface antigen D. So if you contain this surface antigen, since for me, I am A positive, I do carry the surface antigen D on my red blood cells. If I was negative, I would not have that surface antigen D there. So if you look over to that little chart on the right side there, you can see an Rh positive person carries the surface antigen Ds. Rh negative person has no surface antigens here. So here's the thing. Normally, right, a person does not have antibodies to the Rh factor, the anti-D antibodies. They don't normally have those. The only time that a person will gain the anti-D antibodies or the Rh factor antibodies is going to be when they are exposed to an Rh positive a person's blood. So let me repeat that. The only time, because normally the antibodies do not exist. So for those antibodies to come into existence, that individual needs to be exposed to a person that is Rh positive to their blood. Once that happens, then that Rh negative person, right, will produce their own antibodies. All right, so keep in mind. All right, so you can see here, ABO group and Rh type are reported together. Like I said, I am A positive, so I am reporting my ABO blood group and my Rh type there. Right, so as an A positive person, that means I can tell you right now, I have the surface antigen A. I carry the anti-B antibodies in my blood plasma. And I also have the Rh factor or surface antigen D on my red blood cells because I am Rh positive. So keep that in mind. Again, if you're not sure, rewatch the video, watch it a couple times there. So I like this slide here because this slide here throws in, all right, some clinical aspects here to blood typing here. So if you notice on our table, you've got blood type on the left, and then the two columns afterwards, can donate blood to, and then can receive blood from. So this helps us to determine who is safe to donate blood to certain individuals. So for example, I'll go up to me, and you can use this to look at yourself if you know your blood type. I am A positive. This means I can donate blood to another A positive person and a person that is also AB positive. So if we go back to our chart here, I am A positive. All right. I carry these anti-B antibodies, so I do not want to get blood from any individual that has that type of surface, all right, antigen. I do not want to receive blood from type B. If you don't believe me, look here, okay? I can receive blood from these individuals, but you notice B isn't on there. Right? That's because I carry the antibodies for the surface antigen B. So I don't want to get that blood from that individual. And notice what else isn't on there. AB. Why? AB carries surface antigen B. While my antibodies won't react to surface antigen A, they certainly will react to the surface antigen B. So I do not want to get that blood because my own antibodies will interact with that. So type B blood is not good for me, and type AB blood is not good for me. So some of you might be thinking, okay, I understand the B, okay, and I understand the AB, but how come you can donate blood to an AB person? And I'll talk about this. I haven't really quite talked about it yet. All right. I can donate blood to an AB person. And obviously, I can donate blood to a person with the same blood type as myself because they don't carry the anti-B antibodies. So they won't have antibodies for the blood that I give them. All right, because the surface antigens won't match up with the antibodies there. But AB, you go back to the chart, look, if I give that blood to somebody, check it out. That individual does not have any antibodies. So their antibodies will not react to my surface antigens, so it's safe to donate blood to them because they don't have any antibodies to interact with that surface antigen. Right? So when we talk about blood donations here, right, we're always going to consider the recipient's antibody status. Think of it like that. So here comes a situation, all right? If somebody receives what they call a bad blood transfusion, right? An incompatible donor, if they receive, we can get, all right, this process or this occurrence called agglutination. And agglutination is just clumping of blood. That's essentially what will happen. So the person that receives the blood from an individual, their antibodies will start to bind onto those red blood cells that were donated or infused into the individual, and it will start to cause clumping. We call that agglutination. If the clumping is bad enough, it can cause blockage of blood vessels. And when that happens, all right, we're all familiar with certain causes, for example, of a stroke. You can get a stroke from an aneurysm, which is when a blood vessel, for lack of a better term, bursts or tears or whatnot, or you get a blocked blood vessel, which is what we see a lot. This situation can occur. I'm not saying you're going to get a stroke. It can, depending on how bad, but you block some of the blood vessels, and we all know what happens when you're not getting enough blood supply to tissues. The tissues can start to die. Also, the cells can start to break down. We call that hemolysis. They start to rupture. And when the cells start to rupture, those damaged cell fragments can cause damage to the organs and the tissues that they're supplying, but also, if they're not supplying oxygen to those tissues, then you can get tissue necrosis and organ damage there. So that's something that we definitely have to be careful with. All right, so mess around with this chart, kind of look on it and see, all right, what you're thinking. All right, let me see what my blood type is. Who can I receive blood from? Who can I get blood from? So, we look at these two blood types here, A positive and O neg, or O negative. And you can see, all right, O negative is the universal donor. So they can give blood to everybody. And AB positive, they are the universal recipient. Keep in mind, if you are AB, you don't have any antibodies. So you can get blood from everybody. And if you are type O, O negative specifically, look at that. You don't have any surface antigens. Right? So if you don't have any surface antigens, antibodies can't interact with those cells. They don't have anything to latch onto. So now you can understand why we have those terms: universal donor, universal recipient. And now you know. So here's an example here of that erythrocyte agglutination. So you have a type B recipient, which means that that type B recipient carries wait for it, anti-A antibodies. So those anti-A antibodies are floating around in that person's blood plasma. And so now someone that wasn't paying attention just hung a bag, all right, of type A blood. And so guess what? That type A blood has the surface antigen A. Guess who likes surface antigen A? Anti-A antibodies. So they'll start interacting with those antigens and they cause clumping, and then that clumping can cause blockages. And you can see it here on this picture, how we're seeing, all right, so a blockage form. So a quick way to do it, I mean, if you're not sure if someone's blood is compatible with another person's blood is, you will mix the two blood samples together. And if the result looks like this, all right, then you'll say, okay, no clumping is seen, successful match. No agglutination is going to occur. We can actually donate blood to this individual. But if you mix the two blood samples together and you get this, that is clumping. That is hemolysis. It is not a good match. Do not allow the donation, all right, of blood from that individual and vice versa. All right, because you can see the antibody, and in this case here, you got type A, and then you can see the anti-A antibodies reacting to the donor blood because it has the surface antigen A on top, and we get that agglutination and the hemolysis there. All right, and then finally, our first clinical view and the only clinical view for this lab is going to be RH incompatibility and pregnancy. What does this all mean? Remember what I told you, the RH, right, that surface antigen D. An individual will not have antibodies, all right, until they are exposed to an RH positive person. Right? So for example, everyone likes to talk about the RH negative mom. So mom gets pregnant, right? And the fetus there, right, the baby is an RH positive baby. And so, of course, during delivery there is how do I want to say this? A mixture of blood between baby and mom. And so mom is exposed to, all right, that RH positive baby's blood. And now mom's body starts to produce the anti-D antibodies. So now you have to be careful because any future pregnancies, right, especially if the next baby, right, has incompatible blood there, right, some of these antibodies, because they are small enough, they can cross over the placenta there, and the placenta is a great filter to protect baby from a lot of hostile things, all right, in the world, all right, but it doesn't get everything. And so certain, all right, microorganisms and certain molecules can pass over, and antibodies are small enough that they can cross the placenta. And if they do, right, we're going to get that agglutination, we're going to get that hemolysis there, and we start to destroy the baby's red blood cells. That's a problem. So we have a solution for that, and we give pregnant women, right, these immunoglobulins. It's called RhoGAM. Folks, they usually give it a shot to the mom. It's called RhoGAM, and that gives mom these special immunoglobulins, so it helps to prevent this type of situation happening. Because if we start destroying the red blood cells of the baby, you can get what's known as hemolytic disease of the newborn. and the red blood cells in the baby start to break down. And some of the symptoms would be, well, if you think about it, a decrease in red blood cell volume, we call that anemia. So we know all the symptoms of anemia: tiredness, lethargy, all easily bruising easily relatively. But part of this, and we'll talk about it in the lecture, that when we start to break down the red blood cells, your body likes to recycle some of the contents of the red blood cells, and we start to get an increase in what we call bilirubin, which is one of the intermediates of hemoglobin when it's being broken down. So because we're destroying the red blood cells, you start to get an increase in the breakdown of hemoglobin, and so you're going to see an increase of bilirubin, which is one of the intermediates there in that process, and so we'll see a larger amount in the blood. So if you dissect that word, hyper means a lot. Bilirubin is that a specific molecule, then emia is blood. So you will see an increase of bilirubin in the blood. And if it's bad enough, heart failure and death. So again, this is why it's really, really important to know your blood typing. And pretty much now, those immunoglobulins, those RhoGAM shots, are usually given to almost pretty much every mom, regardless if they've had a birth or not. Depends on the hospital policy. And then our final slide for this lab video is a typical blood smear. I don't really have too much to say on this, except use this slide to label any of the structures in your lab manual. There's not really a lot of labeling in the lab manual for this chapter here, but keep in mind, this is not a true representation of a blood smear, because you can see there's a lot of white blood cells here compared to the red blood cells, but we just wanted to kind of show you all the various different types of the blood cells. And I'll go into detail about what these are more so in class, but you can see, right, you have your neutrophils, which are, I labeled these guys right here. All right, these are the most numerous types of white blood cells, and we have several of them located throughout the slide. Right, the nucleus is called it's polymorphular, which is just means you have multilobulated, so you have lots of different lobes to it. Then you can see an eosinophil, right? And that's demarked by the red dots inside of the cell and the shape of the cell's nucleus. Right? Obviously, red blood cells scattered all over the place. We have a basophil. Baso means blue. Eo means red. Okay, and so again, this type of cell has blue dots in it. Again, very unique nucleus. And then we have our monocytes and lymphocytes. The monocytes are the blood's version of a macrophage. Once this cell exits out of the blood, it becomes a macrophage. In the blood, it stays a monocyte, relatively big cell. And then we have our lymphocytes. And you can see the ratio of the size of the nucleus compared to the cell itself. The nucleus takes up a big amount of space inside this cell. And when we get into the immune chapter, you're going to see why, because these cells like to make a lot of things that are going to help kill, right, pathogens and whatnot. And then you can see the smaller cell fragments here throughout, and those are your platelets. All right, folks, I hope you enjoyed this lab video, and I hope you learned quite a bit from it. You folks be safe. Of course, if you have any questions, don't hesitate to contact me. Until our next encounter, please be safe, study a lot, and stay out of trouble. Bye.